Torsten Gross, Charité Berlin, Identifiability and Experimental Design in Perturbation Studies. It's a pleasure to talk to you. My name is Torsten Gross and I work in the Blütgen lab at Charité Berlin. Today I want to address two challenges of network inference problems that are rarely discussed but important. Network inference became really interesting with the onset of the omics age. Omics technologies allow to detect and quantify a huge variety of different biological components, but typically it cannot directly characterize the interactions between these components. Yet exactly these interactions allow us to understand the inner workings of a cell, and this is why network inference methods became so important. A particularly interesting class of those are those that analyze perturbation data. So perturbation data describes how a certain system reacts to perturbations at some of its nodes. So you can think of, for example, gene knockouts, or if you're working on signaling, applying certain inhibitors to certain of the kinases. And so then by looking at the resulting causal chains of reactions, the hope is that these methods are able to reveal functional mechanisms. But as I said before, it is often very far from clear whether a given perturbation dataset actually allows us to uniquely determine a certain link in our network. And additionally, currently we have very little insights into what is the perturbation sequence that delivers us the most information. And so these questions are what I want to ask and address in this work that I'll present to you now. So the point of view that I'll take in this presentation is that of the linear response approach. So these are methods that are based on a specific assumption that I'll explain in a moment. And particularly, I'll take the framework of modular response analysis, which states that you can regard your biological system in terms of a system of ordinary differential equations, which can be arbitrarily complex and nonlinear. And so here, this vector x describes the abundances or the activities of different components in our network. And this parameter vector p, each of its components, describes something that we can modify in our experiments, the perturbations, so to say. And now if you do a perturbation experiment, the idea is that you apply a specific perturbation and then you let the system relax into a new equilibrium state, and then you measure the differences of your new state to the old one. And so here, this is captured in this matrix R. Here what we do is for each of these columns, this represents one of the perturbations, we measure for each of the components in each of these rows the difference between the steady states of the perturbed versus the unperturbed system. And so now taking this information, we're interested in inferring the Jacobian and the sensitivity metrics. So the entries of the Jacobian metrics describe the changes in the rate of, the influence that a specific node has on the rate of change of a specific other node. So basically the influence of one node to another. So this characterizes our network. Whereas the sensitivity metrics describes the influence that a specific perturbation has on a specific node. And now, if we can assume that the steady state function changes linearly with the magnitude of our perturbations, we can derive a relationship between these three quantities that looks like this. But that's not all. Typically, the modular response analysis tries to quantify the edges of networks for which a topology is already known. So what does this mean? Let's take a look at an example. So here we have a four node network. Each of these nodes is represented by one of these circles that gets perturbed by one of these squares here. So these squares, these yellow squares represent the perturbations that can target either one or in this case also two nodes. And now knowing this topology and these targets of the perturbations already constrains our equation quite a bit, and it has a specific structure now, which looks like this. So now here each of these edges in our network is represented by one of these unknown components of the Jacobian matrix, But if we consider, for example, here the entry in the third row in the first column, this would quantify the influence that node 1 has on node 3. But if we look into our topology, we see that there's no link. So we know that this specific component is 0. Similarly, we can look in the sensitivity matrix and see that some of the perturbations do not target certain nodes, and therefore the according entry in the sensitivity matrix is 0 as well. The question that I want to ask now is, does this equation uniquely constrain these unknown entries to a single fixed value, which would mean they are identifiable? Or is it possible, for example, to change a specific entry and then to compensate for this change by changing another entry such that this equation still holds? So this would mean that these two entries are actually non-identifiable. And I think it is one of the central findings in this work that I was able to rigorously show that we can explain or we can clarify this identifiability behavior by looking at the maximum flow problem. So I won't be able to show you the derivation, but I will be able to show you the results of this study. So let's consider again our example here, and let's say we're interested in querying about the identifiability status of all the edges that go into node 3. So the first thing that we have to do is to disregard any perturbation that targets node 3 directly. And then from this reduced network, we construct a flow network. And we do it by introducing a source network up here, which gets connected to all the perturbations, and a sink node, which gets targeted by all of the nodes that directly target node 3. And if you're not familiar with flow networks, really think of these edges as pipes. And we're trying to pump water from the source to the sink node, but we have a constraint, which is that at any node, the maximum flow that can go through must be less than 1. And so now the question is, what is the maximum flow that we can pump from source to sink node? And so in this case, this is 1. Because you can see, so here I colored in red one of the possible paths to which such a flow could go. This is not unique, but you can see that even if we could not increase this flow by, for example, pumping water through this alternative route here, because then in any case there would be more flow than 1 going through this bottleneck node 2. Now coming back to identifiability. Let's suppose we want to clarify identifiability of the edge from going from 2 to 3. What we need to do now is to erase the link from 2 to the source node and then see whether this reduces the maximum flow. So in this case, we see that the maximum flow is not reduced because I can find another path that carries a flow of 1 going to the sink node. And so this means that the according Jacobian entry of this edge is non-identifiable. Similarly, we can ask the question for the edge going from 4 to 3, and then we take out the edge going from 4 to the source node, and once again we see that the maximum flow is not reduced, which means also this edge is non-identifiable. In a similar flavor, we can also ask about the identifiability of the entries of the sensitivity matrix, and then we have to consider a flow network which connects the source node to the according perturbation, and then we know that the sensitivity entry would be identifiable if the maximum flow is not increased. But in this case, we see in our network once again that the entry is non-identifiable because these additional edges would actually increase the maximum flow. In this way, the maximum flow perspective allows us to become clear about the identifiability status of any of our network parameters. And this allows us to go next step, which is to ask about experimental design. So what is the perturbation sequence that gives us the most information about the network? Is it one of these, or is it maybe even one where we are able to combine two perturbations into a single perturbation experiment run? And more specifically, we're asking to find a sequence of perturbations that maximizes the number of identifiable edges. So naively, we could now, using our maximum flow approach, simply enumerate all sequences and simply count the number of identifiable edges. But you will probably agree that this creates a combinatorial explosion because the number of sequences grows super exponentially with the number of perturbations. And so to this end, we implemented a depth-first search on a tree of perturbation sequences that can deal with this combinatorial space and identify the optimal sequences. And so let me demonstrate this at another toy example. So now we look at this network, and I want to distinguish between two scenarios. So in the first case, I want to allow for all the perturbations that target a single node, and in a second scenario, I want to allow for any combination of such single target perturbations into one experimental run. And so if we do that, independent of this strategy, the suggestion is to target, in this case, so the idea is that if you target node 5 in the first, as your first perturbation, you will get 4 identifiable links in this network, which is, compared to all other possible perturbation targets, the maximum number of identifiable links possible. So as a second perturbation, these two strategies suggest different things. So here, if we're doing a single target strategy, it would be to perturb node 1, which would increase our number of identifiable edges again by 1. But in the multi-target approach, it is a better choice to target 2 nodes at the same time, which increases the number of identifiable edges by 1 more edge. And so going on, we see that the number of identifiable edges grows, and after 4 perturbations, the multi-target strategy was able to fully identify, make the network fully identifiable. This is reached in the single target strategy after 5 perturbations. But now if we look into all possible perturbation sequences, we see that if we would have just randomly guessed a perturbation sequence, we would most likely have missed, and we would have needed 6 perturbations in order to reach full identifiability. Now this is only a specific toy example, and so I wanted to test the performance of these strategies on a larger scale. And so I considered all human pathways that I could access from the KEGG database, and I computed, in order to compare the different scores on different networks, a score that I call the Identifiability Area under the curve, and it is simply the sum over the number of identifiable edges over all of these different columns that you see here, normalized by the number of perturbations and number of identifiable edges. So this means that this score would become 1 if a single perturbation is enough in order to make a network fully identifiable, and it would, for example, become 0.5 if your number of identifiable edges grows linearly with the number of perturbations. And so if we compute the score for, or the distribution of scores for random strategies, we obtain this distribution. And then as another control, I also considered a strategy which I call the naive strategy, which is based on the simple idea that I should perturb nodes that are most upstream in a network. The reasoning here is that the more upstream a node is, the more reaction it will show on different nodes, and therefore the more information it will carry. And indeed, we see that the information, or that our score is improved. However, comparing to the single target strategy, which makes use of our maximum flow strategy, even increases our score even more, and even more so with using the multi-target strategy. So investigating the multi-target strategy a bit more, I ask, so what is the typical number of combination or target perturbations that are combined into a single perturbation experiment? And what we see is that the larger the network becomes, the more beneficial it is to combine different single target perturbations into one perturbation experiment. Maybe it might be that we're not so interested in looking at the performance of an entire sequence of perturbations, but all that we want to know is the minimum number of perturbations that is required in order to fully make a network fully identifiable. And so for each of the human KEGG pathways, I considered an ensemble of 100 random perturbation sequences, and I looked at the average number of minimally required perturbations over this ensemble of random sequences, and then I compared this number to the number of required perturbations in the different strategies. And what you see here now is that the number, or that the fraction of required perturbations for the single target strategy is only approximately 1/3, and the multi-target strategy even only 1/4 of that required in the random trial. And so this shows that our optimization algorithm really allows to drastically reduce the required experimental effort. So let me summarize. I've shown you that a maximum flow problem is able to describe network identifiability, and taking this perspective, we were able to implement an algorithm for the optimization of experimental design, and it allowed us to derive sequences, perturbation sequences, that reduce the number of required perturbations by 2/3 or even 3/4, comparing a single and a multi-target strategy, compared to a random control. So if you're interested in trying out these algorithms and tools for yourself, feel free to check out my GitHub. And with this, I would like to thank you for your attention.